Hello, everyone, and thanks for coming to this presentation. So my name is Julio Saez-Rodriguez from Heidelberg University in Germany. And here you can see my conflict of interest. And today I would like to talk about our recent work about knowledge-based machine learning approaches to extract disease mechanisms from multi-omics data. So many of us are interested in finding ways to analyze different types of omics data, proteomic, transcriptomic, and so forth, to better understand disease and ultimately find new therapeutic opportunities. And what we try to do using statistics and machine learning is to find patterns in the data that perhaps are associated with development of the disease, or build more predictive algorithms that allow us to know if, for example, a particular treatment is going to be helpful. Within the scope, what we focus in the lab is how to support these algorithms with the use of biological knowledge. And the key message I would like to convey to you today is that there is a lot of this we know about the molecular process that happens in the cell. And we can use them, as I said, to help the machine learning algorithms by extracting from very large omics data sets where comprises thousands of variables, smaller number of features. And as these features, because they're fewer, allow us to decrease the dimensionality of the problem, therefore increase the statistical power. And also, because they're rooted in well understood biochemical processes, they are better suited for mechanistic understanding. So I will show you some vignettes of this type of approaches, but all the tools that I will show you on Beyond work in a modular way. You can combine in different fashions, and they're freely available in our GitHub repository. The first that we need to do to use prior knowledge is to have access to it. And there are many sources of prior knowledge with different strengths, different emphasis, perhaps cancer context or immunological context. And so over the years, we developed a resource OmniPath that allows you to access many different resources so that you know which ones you should use for a particular type of analysis. So the same way that if you wear glasses, you try different lenses and you pick the one that's the best for watching. With OmniPath, you look at many different resources and then you pick the right ones for your analysis. So over the years, OmniPath have grown as a collection of over 1,000 different resources, 100 different resources covering different aspects of molecular biology, from networks and pathways to complexes to notations and so forth. We put a lot of emphasis in capturing the notation, and so we have at the moment over 2 million different notations, and this is really an effort over the years led by Dinesh Ture. And more recently, in the process of trying to refactor OmniPath in a more modern framework in the context of knowledge graphs, we have, in this work led by Sebastian Loventenser, developed a more general framework to capture knowledge. And this is called BioCipher. And by using harmonization of ontology and other strategies, it allows us to really have a place where you can combine different knowledge from different contexts, from different places, not only molecular, like in our case in OmniPath, but also maybe clinical knowledge or genetic knowledge, like what comes from open targets. So you can use it and maintain it in a better way. And BioCipher is really open for the community, so we welcome participation at all levels. So once we have this knowledge, we use it to extract, as I said, features from omics data. And really, these features can be very different, depending also on the type of data. So for example, if we have transcriptomic data, we can estimate the activity of pathways or of transcription factor. And for this, we develop different resources based on prior knowledge. If you have phosphoproteomic, you can use it to estimate the activity of kinases. And you have metabolomics of metabolic enzymes. But regardless of the omics and the biological processes, what you need to do is to perform some type of enrichment, whereby from the omics data and given the prior knowledge of the components of the process of interest, for example, which are the target genes of particular transcription factor, you do some type of enrichment. So to facilitate this process, Pau Badia in Montpellier developed a tool called decoupler that includes a lot of these different algorithms to perform this type of enrichment, which is super efficient and available both in Bioconductor and in Python. Now, I'd like to discuss in some more detail how we apply these ideas in the context of single-cell RNA, where, of course, there is a lot of opportunities and a lot of excitement. And as you know, a single-cell RNA allows us to measure individual cells, the transcriptomic profile, but it has a number of challenges in terms of coverage of genes, noise, and so forth. So we would like to be able to say, take a group of cells that belong to a particular cell type, like here, cell type B, estimate the activities of pathways and transcription factors, for example. And we performed quite some thorough benchmarks a couple of years ago to convince us that we can use these methods despite the single cell features in terms of coverage and other limitations that I mentioned before. And the idea being is because these signatures are extracted from a group of genes, even if you don't measure them all, if there is some noise, you're able to still obtain meaningful features. So this has allowed us to use them in the context of different disease processes. Something very simple you can do with this type of data is to better cluster them. So if you have, in this case, blood cells, PBMCs, if you would like to cluster them according to the expression of transcription factors, you see here they don't cluster very well, so different colors are different blood cancer types. But if instead you look at the activity of transcription factors, so not looking at transcription factor itself, but to target genes and doing this enrichment, you get a much better separation of cell types. We have used it to characterize, for example, different cell types in kidney fibrosis, also in the context of spatially resolved data, to better characterize multicellular communities in myocardial infarction, and also now more recently in the context of multiple sclerosis, to better understand the disease process. In this case, what you see here is slides from the brain of a patient. If you look at expression of a transcription factor of interest, RFX5, you don't see a very clear pattern, and also you don't measure the transcription factor itself in many places. But if instead you look at the activity estimated from the target of the gene, you see a much more clear picture. And here, where you know that there is the lesion, you see a high level of activity of RX5, which is transcription factor involved in the pathophysiology, so that kind of makes sense. So then this pathway transcription factor methods for single-cell RNA can be very helpful to understand disease processes, and we apply them in many contexts, and we're really happy to discuss this in some more detail. Now, I wanted to discuss in some more detail another strategy where we use knowledge to extract mechanistic insights from single-cell data, which is the study of cell-cell communication. And as you probably know, there is a large number of methods, and what they do is to say, okay, if I know a pair of ligand and receptor, I will look in which cell types the ligand is highly expressed, and then in which cell type the cognate receptor is highly expressed. And if they're very expressed, it's very likely that these two cells are communicating. And this makes sense. But if you think about the molecular processes, there are a lot of things going on and a lot of assumptions we have to make. So what we know is that one cell may express the ligand, but this ligand at the RNA level has to be translated into proteins, and then it has to be secreted. And it has, if it's a cytokine, for example, you have to diffuse and to reach the target cell. And once it reaches it, it will induce the response. So, and all we have is the RNA here in the sending cell and the RNA in the target cell. So it's not really clear why this should work in how well. So to better understand how well these methods work, Daniel Dimitrov in the lab developed a suite called Liana that allows you to run many of these existing methods, because although all methods have like this common hypothesis, they have differences in how they implement the algorithms. They also have differences in which database of ligand and receptor they use. So through OmniPath, we had access to all of them. So yeah, then Daniel tried to benchmark these methods, but it is very hard to have data where you know the two cells are communicating and at the same time have RNA data. So we are always welcoming suggestions for ground truths, but in their absence, we had indirect support by looking if, for example, if you have spatially resolved data, if cells that are more close to each other are more communicating. This is something you would expect and you would like to see in the analysis, or looking at the induction of downstream signaling processes. So we're not going into the details. This is published last year, but just the key point here what we found is that you get different results depending on the method and the resource, although in general, most of them capture some signal. And now, and this is work that is about to be ready, so it depends when you watch this video, this may be already out. Daniel has refactored all this framework, originally in R, now it's in Python, compatible with SCVerse, and have extended the benchmarks together with the open problems communities. It has also other features, for example, the capability to perform analysis across different samples together, multi-sample analysis, also the possibility to better to study communication directly in spatially resolved data. So here, leveraging different tools. MIST is a tool from our lab to study patterns of interactions in space, but there are other tools like SCHOT, Special DM, that we have included in the framework. Also going beyond the pure cell-cell communication to include the enrichment of target processes such as transcription factor activities and so on, to look at the effect not only of protein-mediated signaling, also those mediated by metabolites, and then looking at the intracellular pathways to try to connect the ligand and receptor to the transcription factors by looking at intermediate pathways. Just as an example of how this would work and how this links also to the earlier analysis I showed before. As I said, we can perform enrichment on pathways, and by looking at the same time at the presence of cell-cell communication events and their co-localization with transcription factor events, in this case using an adaptation from SCHOT, the method here developed by Sheila Gazafar and colleagues, we can start to look in space if there are relationships between cell-cell communication events and transcription factor activities. So that's a bit of an overview of the methods that we have for analyzing single cell and spatial data, from pathways, transcription factors to cell-cell communication. And I really welcome you to look more into our work and again to reach out to us if that's of interest. That's in the last couple of minutes. I wanted to bring up something else. And this is something that also we are many in the community interested in, and there is a lot of also activity in this conference, is to better understand how we can leverage large language models. And I think most of us at this point are familiar with the fact that they are really powerful tools that can remember a lot of information, right? They're trained on large data sets, but also that they have limitations, in particular the lack of awareness and these logic deficits and this tendency to hallucinate, to basically to always give us an answer, even though there is not a real answer. So it's often hard to know if what we query one of these models, whether the answer is real or not. And there are many uses and many people working on this. As I said, in our case, we are trying to understand how we can use these approaches downstream of the type of analysis I described. So imagine you have some omics data, you have done some enrichment, some of these pathway transcription factor and so forth. So you could then envision coming with those results into one of these large language models and ask it questions about your data. And so again, Sebastian Loventasser has built a model which is early days, and we are very happy to work with others. Here you see the link, where not only you have the option to query your LLM, and it's modular, you can use any of the existing ones, but also having like a complementary loop where you use the things I showed you at the beginning of the talk. So this knowledge that we can access from BioCipher to kind of give the context to the query. Also use specific papers to even more constrain the framework and use it as a fast checking mechanisms, and in this way interact with the researcher. So this is early days, but really, if you took samples, this is one from student Heidelberg, Sergio Gomez Ochoa, who works on heart problems. So he had some differential express genes in the context of a particular heart disease, and asked to the chat to explain a bit the data. Not only can they give you a bit of background of what is known about the genes and what they can mean, but also you can ask it, for example, to give you some visual representation and some contextualization of the results. And again, this is preliminary days, but I just wanted to mention that we're interested in this, and I'm really happy with others who want to address this type of problems. So that's all I wanted to say today. I'd like to thank the people in the lab who did the work. I mentioned them along the way. We have a number of great collaborators, both experimentally and computationally, and these are our funders. So yes, I will leave you with this idea that we think that you can use biological knowledge to help you to better understand omics data in the context of human disease and drug development, can give you better candidates where you can now try to target with different therapies. And by doing so, we hope to be able to better progress in finding new therapies. With that, thank you for listening. And if not online, I'm happy to take any questions by our means. Thank you.